cholesterol synthesis. Now, cholesterol is an important compound in the body which is the structural component of cell membrane. Its function is to maintain the fluidity of cell membrane whether in hot or cold temperature. It acts as a precursor of bile acid and steroid hormones including vitamin D. Now, we're going to talk about the synthesis of cholesterol. So the synthesis of cholesterol involves three steps: the synthesis of HMG-CoA, the synthesis of mevalonate, and the synthesis of cholesterol. Now, explanation. First of all, we're going to talk about the synthesis of HMG-CoA. So cholesterol synthesis starts with the combining of acetyl-CoA molecule. Two acetyl-CoA molecules combine together where by the action of thiolase enzyme a molecule of CoA is released and acetoacetyl-CoA is formed. Now another molecule of acetyl-CoA is added in this acetyl-CoA to acetoacetyl-CoA to form a compound containing 3-acetyl-CoA known as the 3-hydroxy 3-methyl-glutaryl-CoA, or in short we can say that HMG-CoA. Just like that, since it has only one CoA, so acetoacetyl is added and CoA is released. This reaction occurs in presence of HMG-CoA synthase. Now we have formed the HMG-CoA, so the first step is completed. Now the second step is the synthesis of mevalonate. Now mevalonate, when HMG-CoA is reduced by the action of HMG-CoA reductase, the CoA is released and mevalonate is formed. Since it is a reduction step, so NADPH is utilized in this process. So its hydrogen are added to HMG-CoA, and since CoA is released from this, so this reaction is irreversible. Another importance of this reaction is that it is irreversible reaction along with the rate limiting step. Since it is a rate limiting step, so HMG-CoA must be tightly regulated. Regulation of sterol includes the sterol regulatory element binding protein. This is present inside the endoplasmic reticulum combined with the cleavage activating protein of this one and the INSIG protein, which is a gene product of insulin action. Now, when the cholesterol level is low, when there is low cholesterol level in the body, the INSIG gene separates from this complex. So this complex that is catalyzing, that is cleavage activating protein and this sterol related element binding protein. This complex is released from the endoplasmic reticulum. Here is the protein complex which is released from the endoplasmic reticulum. Now this complex moves toward the Golgi apparatus where proteolytic cleavage of the SREBP occurs. So this single SREBP moves inside the nucleus and binds with the sterol related element on the DNA, which initiates the transcription and translation and causes the production of HMG-CoA reductase. Now another factor includes the phosphorylation factor. When a kinase, that is AMP dependent kinase, act upon HMG-CoA, it is phosphorylated and goes to its inactive form. While when a phosphatase acts upon it, this phosphate is removed and the HMG-CoA reductase moves towards its active form. So upon high cholesterol level, it decreases the HMG-CoA reductase, and low cholesterol level, it increases the HMG-CoA reductase activity. Other regulatory factors include the insulin and tyrosine, both of which are the enzyme of well-fed state. So they upregulates the HMG-CoA reductase. While in case of starvation or fasting, glucagon and the steroid glucocorticoid are released, so they downregulates the HMG-CoA reductase. Now what happens if the cholesterol level is high? with the direct action of the insect with the HMG-CoA. Now HMG-CoA is also cholesterol sensitive. When it senses that there is high cholesterol level, it directly binds with the insect protein and undergo proteasomal degradation. Now talking about the synthesis of cholesterol, we have made in the first two steps up to mevalonate, which is a 6 carbon compound. Now mevalonate is acted upon by a kinase which adds the phosphate in it and converts it into a 6 carbon compound known as 5-pyrophosphate mevalonate. 5-pyrophosphate mevalonate. Now since it's a 6 carbon compound, when it undergoes decarboxylation by a decarboxylase enzyme, it is converted into 6 to 5 carbon compound. One carbon is removed as carbon dioxide. So from pyrophosphate mevalonate, it converts into isopentenyl pyrophosphate, which is a 5 carbon compound. Now this compound undergoes isomerization, which is reversible on the action of isomerase enzyme, and converts into dimethyl pyrophosphate, which is also a 5 carbon compound. Now the dimethyl pyrophosphate and the isopentenyl pyrophosphate, both of these combine together, undergo condensation and releases 2-phosphate molecule on the action of transferase enzyme to get converted into 5 plus 5 10 carbon compound, which is known as geranyl pyrophosphate. Now this geranyl pyrophosphate further undergoes condensation. How does it undergoes condensation? This 10 carbon geranyl pyrophosphate is added up by the 5 carbon isopentenyl pyrophosphate, so it makes a total of 15 carbon. 2 phosphate groups are released, so the it converts into ferrenyl pyrophosphate, which is a 15 carbon compound. Now this ferrenyl pyrophosphate is acted upon by an enzyme called squalene synthase. 2 phosphate groups are removed from it and the reduction of it occurs to convert this 15 carbon compound, which is added upon by another 15 carbon compound ferrenyl pyrophosphate, to form a total of 30 carbon compound known as squalene. Now this 30 carbon compound squalene is the last linear compound in cholesterol synthesis. Now as I have said, it is the last one, so the compound next to it will be ring structure. So this squalene compound undergoes a series of reaction in which the oxygenation occurs, the reduction occurs, and cyclization occurs to convert it into a ring compound known as Lanosterol. Lanosterol, just like it, is a 30 carbon compound and the first ring sterol in the synthesis process. Now this Lanosterol undergo a number of reactions, 17 to 19 reactions, in which it undergoes chain shortening, that is decreasing the number of carbon and decreasing the number of double bond, to get converted into dehydrocholesterol. If we recall from the chapter of vitamin, we have studied that even a single photon of which act upon the dehydrocholesterol can convert it into vitamin D. So it is a precursor of vitamin D. It is also a precursor of bile salt, bile acid. Okay. So when this dehydrocholesterol is reduced by 7-dehydrocholesterol reductase, it gets converted into cholesterol, which is a 27 carbon compound. Now we have discussed the synthesis of cholesterol. Now talking about the excretion of cholesterol. Now cholesterol, we cannot degrade cholesterol like normal compound, that is we cannot break down cholesterol into carbon dioxide and water. So cholesterol is excreted via two processes, that is conversion to bile salt, bile acid, and also it is directly secreted into the bile. Both of these move to the intestine and finally released from the body as fecal material. So giving a quick summary of the process, The cholesterol synthesis consists of synthesis of HMG-CoA. We have made HMG-CoA by the condensation of acetyl-CoA. Next, the synthesis of mevalonate, which we have formed by the reduction of HMG-CoA, and then the synthesis of cholesterol, which includes the kinase action, decarboxylation and condensation, all of which result in the formation of squalene, which is a linear compound, and then we do the cyclation of the squalene compound to convert it into lanosterol, and lanosterol moves toward the series of reaction to form 7-dehydrocholesterol, and finally reduction provides us the cholesterol. So that was all for today's lecture. Please subscribe the channel and thank you.
Yep.